So the lymphatic system is in the mammals, it's a network of blind-ended lymph capillaries, muscularized collecting vessels, and ducts. So you start off here as the lymph capillaries. They become pre-collectors. They become then the muscularized lymphatic collectors that are divided up into little units separated by valves called lymphangions. And includes also the lymphoid organs such as nodes, tonsils, and Peyer's patches. It's a semi-open, ended, one-way transportation system for fluid, macromolecules, particulate matter, and cells to move from the interstitial spaces through this network all the way through the nodes and eventually be dumped back into the blood into the junction with the subclavian veins. And as I was saying before, it has critical roles in fluid macromolecular homeostasis, inflammation, edema resolution, kind of its classic ideas, lipid uptake, and immune cell antigen trafficking. All of those require transport of the substances from the tissue spaces through the network into the blood. And mammals, because the pressure gradients that exist for the lymphatic system aren't like the heart and the blood vascular system, they don't predominate movement of fluids in a passive way. And even though many people, particularly oncologists, talk about lymph drainage patterns, the reality is lymph drainage implies that there's a passive pressure gradient that moves that. That doesn't exist in 90% of the tissues, 90% of the time. Instead, you have to use active mechanisms, this pumping approach, and the valves to overcome the adverse pressure conditions that normally exist. And that fluid homeostasis plays a critical role. So this is the classic idea of microcirculation, where this is the microcirculatory compartment. This is a capillary with blood flow in it, and you have pressures that across that capillary between the blood and the interstitium, and likewise that lead to fluid and macromolecular movement out of the blood into the interstitium to serve those cells. Some of that fluid will be returned into the venous part of the circulation, but actually not very much less than we teach you in medical school. Most of it, in fact, gets returned back to the blood by going through the lymphatic network. And you can use a similar kind of concentrations and pressure gradients to determine what fluid flows come from the interstitium into the lymph and then move along that lymphatic pathway. And this is just a quick pressure gradient to show you that the pressures in the lymphatic system, number one, are very low compared to blood pressures. So we're talking about things that are near atmospheric, near zero, in fact, up to maybe 7 or 8 centimeters of water, where you have to return it back to the jugular vein. So what that means is you have to move lymph along this pathway, because these are organized from the most peripheral to the most central, because all these go into either the thoracic duct, which drains the whole lower half of the body in the upper left quadrant, or the right lymphatic duct, which drains the upper right quadrant and empties into the jugular veins. And the pressure gradients is in the direction opposite for what passive flow would call it. And so lymph doesn't drain down a hill. In fact, it needs to be pushed or propelled uphill. And the analogy I use is that because my colleagues that work in the blood vascular system often tease me that I work in the body's sewer system. And to some degree that's true. However, it's not quite as simple as sewer systems in this city. It's more like the sewer system in New Orleans, where the net level is below sea level of the city, and so you have to use pumps and valves in order to be able to keep that city from getting flooded. That's exactly what goes on with the lymphatic system. Now, mammals don't have specialized lymphatic hearts like lower vertebrates, like frogs, amphibians, reptiles, and some fish have to move lymph. Instead, we use the networks of pumps and valves to overcome these pressure gradients, and the lymph pumps are either intrinsic, meaning that that pumping mechanism resides in the lymphatic vessel itself, or it's extrinsic, such as skeletal muscle contraction. Every time you exercise, lymph flow, for instance, in my bicep is going to go up dramatically, 10, 20, 50-fold by exercising, because you compress and decompress these very, very flaccid, highly compliant lymphatic systems that are tightly enmeshed in the surrounding tissues. And that, when you couple that with the valves, is basically working like a primer valve. And you're moving fluid to the next little section, or the next little lymphangion, opening and closing valves appropriately with the right types of pressure gradients, as either you generate pressures locally by intrinsic contractions or externally by extrinsic compression of skeletal muscle, heart, GI peristalsis, lung movement, etc. And so when we study the function and dysfunction of the lymphatic system, what our lab group has focused on for the last 20 or so years has been looking at the functions of the pumps and the valves within this system. And here's an example of one of these lymphatics. This is actually a very small lymphatic, about the diameter of your hair. This is the upper edge of the wall. This is the lower edge of the wall. This is an intravital microscopy image. You can see the contractions. This is real time and relaxations. You can kind of see the shadows of the muscle cells wrapping around like fingers on that vessel. This is in the rat mesentery. about 100 microns or so at rest. And here's some blood vessels that metabolically serve that lymphatic because it's actually very, very metabolically active. You can occasionally you'll see the immune cells come zipping by. There's a few that are freeze-framed that come zipping by from right to left here. And so this is what the vessels look like in situ, and you can see that they act both as a vascular system, and in this case, the intrinsic pump acts as a heart itself, driving its own fluid movement. Here's an example of one. This is actually mislabeled. This is a cervical lymphatic, and it's about 300 microns in diameter. I changed the video, but I forgot to change this part of the slide. And I put a cervical vessel in here because this is a unique part of the body that normally can use gravitational forces when you're standing upright to move lymph along this low pressure gradient because it's above the heart. But in fact, it not only uses the gravitational forces, but it still has its pumping activity. And it's pertinent to what we were talking about at the end of my talk here as well. This is one that we've taken out of the tissue, isolated, pressurized, cannulated, and then studies its function. Sure.
Is the intrinsic pumping rate different in different parts of the body?
Yes. And in fact, what we have in this vessel, in the cervical vessel, is a relatively high frequency, low amplitude pump, and it's not terribly responsive to some of the things that regulate the pumping frequencies elsewhere that I'll talk about in just a minute. And so it's kind of a low level, constant pump going on and on and on. Whereas if you go down to your feet, the vessels there are very responsive to changes in pressure. So as you increase the pressure and you stretch them, they pump harder, they pump faster, and basically follow Starling's law of the heart, but for the lymphatic heart instead. And so you're right, there's heterogeneity in the different parts of the lymphatic vascular system, just like there is in the blood system. And we analyze these by looking at, we measure the diameter over time microscopically and then do some estimations, and we can look at the pumping characteristics. So we use basically cardiac analogies, contraction frequency, ejection fraction, pump flow, fractional or real pump flow. And we can also analyze, because there still are a vascular system, we can analyze things like basal tone and compliance of these vessels. They have very low tone, very high compliance, and the contraction frequencies on most vessels that we study in rodents are in the range of 4 to about 20 contractions per minute. That goes up a bit in humans and larger species simply because they have to drive more fluid up a greater hydrostatic pressure gradient. So what are some of the things that intrinsically regulate this? Well, we have, as I indicated, the intrinsic factors, and some of those are what we spend most of our time in my lab studying. That is, there's physical hydrodynamic factors that we know affect blood vessel function. They also affect lymphatic function. So if pressure and stretch is one that's been known since really the 50s to be a driver of lymphatic pump activity. So the more you increase the pressure or stretch that vessel, it will pump faster and it will pump harder to a degree before it begins to fail, just like a heart does. Flow and shear also affect it because as lymph moves along there, it rubs on the lymphatic endothelium because it has an endothelium somewhat similar to blood vessels, somewhat different. And that endothelium is very, very sensitive to shear at magnitudes probably an order and a half lower than what blood vessel endothelium is. And it responds to an increased flow or shear stress acting on those cells by producing nitric oxide, prostanoids, and even to some degree histamine that will then regulate that pump by activating or deactivating the smooth muscle, lymphatic smooth muscle surrounding them. That's also regulated by chemical factors released by the lymphatics itself. I just gave you one example of a paracrine factor. I gave you a couple examples, prostanoids and nitric oxide produced by the endothelium in response to different physical and other chemical activators that will then feed back onto the lymphatic muscle and modulate its function. Substance P we found to be a very powerful neuroinflammatory agent is also a very, very potent stimulator of contractile activity within these vessels. And various cytokines can either activate them or inhibit them depending on what's going on. Growth factors, like VEGF, for instance, potentially can modulate that. There's some indications in the literature that VEGF can stimulate some of these lymphatic vessels in terms of transport characteristics. We haven't been able to recapitulate that, and what we see is that VEGF acts as its old nomenclature was VPF, vascular permeability factor, and makes these vessels inherently much more leaky. So VEGF-C is one that has been reported to activate this. We don't know how much VEGF-C modulates the permeability. It seems to have a minimal effect compared to VEGF-B or VEGF-A. Extrinsic fact, because these lymphatics, in particular lymphatic endothelium, has a different characteristics of VEGF receptor. They have a lot of VEGF-R3, which is more responsive to VEGF-C and VEGF-D, or placental growth factor, than VEGF-B or A. Extrinsic factors can also influence it. Neural factors are some of the classic means of modulating lymphatic function. The adrenergics, epinephrine, norepinephrine, will tend to stimulate this contractile pumping activity and increase lymph flow, at least transiently. Cholinergics appear to have just the opposite, and acetylcholine probably works through nitric oxide release from the endothelium to inhibit it. Substance P already indicated as a powerful stimulator of it, more so of the tonic constriction than this phasic pumping activity. And there are other circulating factors that can activate it or inhibit it, depending on the case may be. Many of those are inflammatory products, so oxygen radicals and cytokines, chemokines. Most of those have been tended to inhibit this activity. Some of them actually do stimulate it, however. The last little bit of a primer I'm going to give you on lymphatic function is that even though I've spent most of my career studying its ability to transport fluid and thus modulate edema and edema formation and edema resolution, in fact, as we found in the last 6 to 7 years, it may really be designed to use that fluid movement to carry immune-relevant agents. And if you look at it anatomically, so if you look at the anatomically at the cardiovascular, blood cardiovascular system, the center of that is either the heart, if you're a cardiologist, or the parenchyma, if you're a cell in your body. And everything goes to and from those different areas. If you look at the center of the lymphatic system anatomically, what its center is the node. You carry things from the parenchyma to the node. You carry things from the node back to the blood to be circulated back to the parenchyma. And so I have changed my stripes, if you will, in that I believe the lymphatic system is actually teleologically and evolutionarily designed to carry things for immunity to and from the nodes. It does so by carrying fluid, and it plays an important role in edema prevention and resolution, et cetera. But its principal cause is probably to do this. I won't spend a lot of time on our more recent work in looking at some of these things, but some of the classic roles, one of those that most immunologists know is that the lymphatic system is critical for the transport of antigens from the interstitium to the nodes. They are the principal route of transport of all large macromolecules, particles, cell debris from the interstitium to the node because it's designed to do that. The blood vascular system is not designed for it. So anything above about 5,000 molecular weight will predominantly be transported greater than 90% by lymphatics versus blood. It plays a key role in this process. It also plays an important role in trafficking immune cells, particularly the professional antigen-presenting cells that Betsy was talking about a little bit earlier, that and in some cases MHC2 or MHC1 from in the immune system, such as dendritic cells, Langerhans cells, macrophages, and B cells, both from the tissues to the node, and in this case from the nodes back to the blood, because it's also important for trafficking the lymphocytes. Hence, that's where the name became, because lymphocytes are predominantly carried from the node back to the blood via lymphatic system. One of the things that we've found, so the lymphatic plays a key role in this process, particularly from antigen presentation for the reasons I gave you. It's the principal route by which most macromolecules from the interstitium are picked up and hence carried back to the node and eventually back to the blood. And so most antigens are fairly large molecules, and they will be predominantly carried via this system back to the node. So we know it plays a key role in that. And when you look at the antigen presentation, so if I get a mosquito bite in my toe, and we look at the foreign antigens produced by that mosquito bite, they'll be picked up locally by macrophages and dendritic cells, and those cells will slowly make their way to the lymphatics and then go back to the node, but that takes hours. Many hours, in fact. However, if you look at that antigen, you're going to see that antigen appear in your popliteal node behind your knee in about 10 minutes. So it's orders of magnitude quicker. And so that means that that antigen gets processed and presented to the cells in your lymph node within a very, very short period of time. That's been known actually for a fairly long time, even though most immunologists don't want to accept that. Some do. What we also have found out recently is that the prenodal lymphatic vessels, including the muscularized lymphatics, having a very extensive coverage of MHC2 positive cells. And these MHC2 positive cells pick up that antigen carried in lymph in a matter of minutes and appear to move within less than an hour. Now, the problem we have is we don't know exactly where they move to. It looks like some of them may go to the node. It looks like some of them may leave the vessel and go to the parenchyma. And our colleagues and I are having extensive collaborative discussions and debates about exactly where they go. But they play a very critical role, and so there's a relatively new paradigm in which these things move and change. Okay, I'm getting the 10-minute notice, so I'm going to move forward. So the problem is, what we do teach medical students is that there are two tissues that normally don't contain lymphatic vessels, bone and brain. So what the heck am I doing here talking to a bunch of neuro-oncologists? Well, there's a number of reasons why I want to talk about this and why I think it could be important to you, and I'm going to try to convince you of them and then let you think about it at the end. One, glioblastoma patients have increased leaky peritoneal blood vessels. That makes the movement of fluid out of the blood into the brain more abundant. You get peritumoral edema, elevated intracranial pressures that produces many of the symptoms that your patients present to you. Secondly, the tumor can obstruct or at least increase resistance to the normal central cerebral spinal fluid pathways. And the last thing that you probably don't know and most people don't appreciate is that lymphatics have recently been shown in the last 10, 15 years to play a very, very critical role in the normal regulation of CSF pressure and fluid flow, even within the CSF, even though lymphatics don't live in the CNS proper. And so the traditional view of CSF transport, I'll go over quickly since you all should know this, is that produced by the choroid plexus, volumes are there, and typically you were taught that CSF is primarily absorbed across the arachnoid villi granulations into the veins of the cranium. And that's true. What you haven't been taught about is on the next slide. It's also low protein and fairly low pressures. Although if you look down here, most of those are going to be higher pressures than what you see in any cervical lymphatic systems that we'll talk about in a second. And so this is the process by which it happens. All the arrows are showing fluid movement in pathways, and here's where the exit of CSF is normally thought to occur as traditionally taught. Well, what's been shown over the last 15 to 20 years is that actually that's true, but the role of extracranial lymphatics plays just as important role. In fact, the amount of CSF absorbed by extracranial lymphatics is about the same as absorbed by those arachnoid villi and granulations. Even though the CNS doesn't have it, the people have demonstrated an important link between interstitial fluid, CSF, and extracranial lymph. And that when they started doing tracer studies, doing macromolecular tracer studies, what they see is that about half of that tracer will leave the CSF via lymphatic system. So it's not a tiny amount. It's a very, very substantial amount. And typically what they do is they leave the cranium along extensions of the subarachnoid space that are associated with most cranial nerves and even some spinal nerves. And in doing so, they get juxtapositioned near a very extensive lymphatic plexus that I'll show you in just a second, which is poised at exactly the right spot to pick up all that fluid coming out of the CSF. And one of the ones that's been studied the best is the cribriform plate lymphatic plexus that lies right below the cribriform plate in basically the nasal mucosa. The olfactory nerves penetrate this cribriform plate through holes or foramen and terminating in olfactory epithelium, and CSF flows along that subarachnoid space with those nerves across that cribriform plate and is absorbed by the lymphatic plexus that exists right there in that nasal mucosa. And so the nasal mucosal lymphatics pick up a lot of things from your nose, but it also collects a substantial amount of its lymph is coming from CSF. It's been demonstrated in rodents, sheep, pigs, humans, and non-human primates. And in some of those, these drain, in some of the lower species, like rodents, it actually drains almost connected exactly with the lymphatics. That is, the lymphatics open right up to those subarachnoid spaces next to the nerves. And so in addition to this classic one, you have this mechanism by which a significant amount of fluid, antigens, are drained via lymphatics into the lymph nodes in the head and neck, and then into great veins of the neck. And so these systems may be important to looking at all the changes that you see in intracranial pressure associated with glioblastoma. And in fact, may be important to the ideas of antigen presentation in glioblastoma and cell movement in glioblastoma. None of that has ever been studied. Someone have a question here?
When you say in the last 10, 15 years there's been a paradigm shift or understanding, is that from experimental work in the lab?
This is primarily from experimental work by Miles Johnston at the University of Toronto. He took some of the old data where people showed tracer studies, injected into the ventricles of the brain, and showed that most of those tracers end up in the cervical lymph nodes and retropharyngeal lymph nodes. And then said, well, if that's the case, we know that most of those pathways only go through lymph, not through blood. And I know Niles very well, he's retired now, but he went through hell getting it accepted. In fact, it drove him into retirement. But it's get accepted now that he's retired, fortunately. And I won't go over all these, but this is basically a number of mechanisms that that he and others, including some of what we've been working on, have shown to actually demonstrate that that pathway accounts for at least 50% of CSF reabsorption from the brain under normal conditions. And under hydrocephaly, if it's experimental hydrocephaly, it accounts for even more. Now, what we don't know, and so let me skip through that. And so here's what we're talking about. Here's the nasal septum. Here's the nasal passages. This is the olfactory lobe. Here's the cribriform plate right there. And then right there are the ethmoid turbinate bones that have the nasal mucosa that have this extremely extensive lymphatic plexus that I'll show you in just a second. This is an example of one of Dr. Johnston's early work where he showed the tracer, which is this substance here, being taken up into what he presumed was lymphatic vessels, but in fact didn't show by immunohistochemistry was lymphatic vessels at that point. Then later on, he went and showed that by this staining to show that this is actually lymphatic vessels here. Here's the olfactory nerve. Here's the cribriform plate. And you see there's a blood vessel right there. And he used two different stains to stain the lymphatic vessels versus blood vessels and showed that a lot of that stain that comes out of the ventricular CSF system is actually predominantly taken up only by the lymphatics and not by the blood. And here's an example in rat, rabbit, sheep, and human of the extensive lymphatic microfill. These were microfill injected into the ventricles and then taken after death, and even shortly after death, looking at all the lymphatic vessels that are extensively associated with the olfactory nerves right below the cribriform plate. Also, all this yellow are lymphatic vessels that have taken up a dye injected into the CSF. So it has a very, very high prevalence of importance. And so now the schematic that is now beginning to be accepted is that there's both means. You have the subarachnoid villi and granulations absorbing it, and you have the lymphatic plexus absorbing it, not only at the cribriform plate, but also along the spinal nerves and elsewhere. So in summary, what we know is the lymphatic reside immediately outside the CNS are important for the component of CSF resorption paths and intracranial pressure regulation. That's been pretty well established now. There's, by this time, close to 75, 80 papers on this. And this role for lymph uptake and removal of CSF may become more critical during periods of elevated intracranial pressure. We know that from experimental methods. What we don't know is that true also in pathological methods like glioblastoma? Oops. So some provocative questions for you all to think about is what happens to CSF removal via lymph flow during the development of glioblastoma and the increases in intracranial pressure that occur. What happens to the macromolecular flux, most of which can be antigens, also coming from the CNS via going to the lymph nodes and stimulating the immune process that Betsy was talking about. And since lymph provides the main pathway for the transport of immune cells, particularly the innate and antigen-presenting cells, from tissues to the lymph nodes, what happens to that immune cell flux under these conditions? In most cases, even though we know normally lymph flow via this pathway goes up with intracranial pressures, we in fact don't know that during glioblastoma because of the changing conditions that are going on there. So it would be, unfortunately, very difficult to do in human patients. You'd have to develop some model to be able to do that. And lastly, can the lymph outflow from CNF be manipulated or increased to at least help ease in a palliative way some of the symptoms that are associated with intracranial pressure elevations and the problems associated with using dexamethasone and other corticosteroids. It's something interesting to think about, and I'll leave that for you guys to ponder. Meanwhile, these are the folks who really did almost all the work, and most within our department, except for an outlier here, Karen, but she's always an outlier, a very nice outlier. And then some folks that were trainees, and there is a Zavea connection in our laboratory as well. And supported by a number of grants from NIH and NASA. And I'll be pleased to try to answer questions if I have a minute. Quick questions.